Hello, dear colleagues. I'm Rustem Sayakhov. I'm president of MultiCASE Incorporated, and today I would like to welcome you to our webinar, which is titled Predictive Toxicology Tools for Confident Risk Assessment. I believe it's going to be a very exciting webinar, and it will be presented by Dr. Crystal Race, who is our senior research scientist. Crystal earned her Bachelor in Science in Chemistry and Mathematics from Altman College in Michigan, and then she got her PhD from Yale University. She is heavily involved with computational method to model proteins, bioorganic systems, and drug bindings, and she is very well versed with classical simulation and quantum mechanics calculations. Crystal joined us in beginning of 2024 as a scientist, and she is already a senior scientist. And she has been working on to build new statistical models to predict toxicological and degradation endpoints, as well as teaching new users how to use the software and how to interpret those reports. Besides this webinar, you will have some other opportunities to meet with us and talk with us. These are presented on this slide. We'll be at a ACT conference, usual, which is this time happens in Phoenix, Arizona. That's going to be in November, second half of November. Before that, we'll be going to China at IUTOX, International College of Toxicology. It's going to be in Beijing in October. And November will be finished for me by going to GMS meeting in Shizuoka, Japan, in around Thanksgiving. It's going to be one interesting piece of our webinar, which will launch right after my presentation here. It's going to be a quick poll, really quick, 1 minute or so. If you can, please answer those simple questions and submit your response. It will not take much of time, but it will help us to understand our audience, and in the end, it will benefit all of us. With this, I'm passing the floor to Dr. Crystal Race for her presentation. Thank you very much.
Hi everyone, thank you for joining us today and welcome to the webinar. My name is Crystal Reese and I'm a senior research scientist here at MultiCASE. So the goal of this webinar today is to give you kind of a general introduction to MultiCASE, give you a bit of our history and our priorities, and then I'm also going to take you through our three software platforms, those being Case Ultra, Meta Ultra, and QSAR Flex. And then for QSAR Flex and Case Ultra, I'm also going to go through some of the individual endpoints that are part of those software packages, including some case studies within the endpoints. And then I'm going to discuss a little bit about what's coming up next as far as new products that we have in the works. And then finally, I'm going to finish up with some follow-up information for how you can get in touch with us for setting up a trial or a demo, and then opening up the floor for some questions. So MultiCASE, we're currently a team 10 strong. We're led by Dr. Rustem Sayakhov and Dr. Suman Chakravarti. Gianna is our VP of Customer Relations and Sales, and she, along with Shannon, are usually going to be your contact people for setting up trials, demos, or getting a quote for our software. Kate is our VP of Operations and Business Development. Munika is our Senior Database Developer. Radha is our Senior Software Developer, and she works alongside Shri, who joined us just earlier this year. And last but not least is Melissa, who is our Senior Operations and Marketing Coordinator. So first, a little bit of the history of MultiCASE itself. So we were founded in 1995, meaning that this year we're celebrating our 30th anniversary. However, our history actually extends about 11 years even farther back to 1984, when our founder, Dr. Klopman, developed the first fully automated and data-driven QSAR modeling technique. And he also released our first commercial software case that same year. Since then, our technology has been adopted by industry, academia, and government agencies, and that includes the US FDA, which has been using our technology since the 1990s. Over the past 20 years, we've had a number of research collaborative agreements with the US FDA in support of the use of predictive technology in regulatory science, and that is ongoing to this day, where we have several ongoing RCAs with the FDA, including ones with CEDAR, CF-SAN, and CVM. At MultiCASE, we have a few guiding principles that direct us in how we actually develop our products. So the first is that we believe that in order to develop the best products, we have to heavily invest in research and development. We believe that those products should be based on techniques that have broad applicability, meaning that they can adapt to a wide variety of different endpoints with minimal to no adjustments needed between those different endpoints. We believe that the products should produce clutter-free and transparent QSAR outcomes to maximize the readability and interpretability of our reports for our customers. And while we hope that our reports are very clear and concise and easy to read, we do understand that sometimes you will have questions. And as part of that, we strive to provide the best customer support. And as evidenced by our ongoing RCAs with the FDA, we believe in supporting use of in silico approaches in regulatory science. So how do we accomplish these goals? So all of our softwares are based on this dual structure where we combine these carefully curated databases with validated predictive algorithms. Many of our software's reports will also provide analogs for those predictions, meaning that our predictions are supported by chemicals that have experimental data, along with the literature sources for that data. And then we also strive to make our alerts as interpretable as possible. In the case of mechanistic alerts, that means that we're providing the mechanistic explanation for why this particular alert is considered to contribute to toxicity. In the case of statistical alerts, we're explaining numerically how this specific alert contributes to that endpoint. So clearly, we very strongly believe in predictive toxicology, and there's a few different reasons for that. So first of all, predictive toxicology reduces the need for animal studies. It's unlikely in the near future that we'll be able to completely eliminate the need for animal studies. However, by leveraging the existing data, both for in vivo and in vitro studies, we can use that data to make predictions for new chemicals instead of needing to perform more animal studies. Additionally, computational methods are also going to be much lower cost and higher throughput than lab-based techniques, with animal studies ranging into the millions of dollars, whereas our tests usually run several orders of magnitude less than that. And our tests also will be able to run in a few seconds to maybe minutes, depending on how many chemicals you're running at the same time. And we also allow you to test multiple endpoints simultaneously. And lastly, again, with this being a computational approach where we're showing you which alerts are contributing to that endpoint, we're showing you exactly which functional groups are going to be toxic, which can aid in drug design. And for the expert rule-based models, we're also providing the mechanistic explanation for why those functional groups are problematic. Most of our models will also provide you with physicochemical property predictions for those same compounds. And then most of our reports will also provide similar analogs that do have that experimental data, as well as literature references for you to go and read more into if you would like. Now, computational toxicology is applicable across a wide range of real-world applications. This includes impurity assessments for pharma and medical devices, ingredient safety reviews for cosmetics and other consumer goods, as well as regulatory screening for industrial and agricultural chemicals. Currently, we have 3 software platforms available. The first is our flagship software, QSAR Ultra, which was released in 2011. QSAR Ultra provides binary predictions for quite a few different endpoints, including mutagenicity, several different systemic toxicities, developmental toxicity, skin and eye, and quite a few more. QSAR Ultra also allows you to perform analog searches for your compound to aid in read across. These analog searches can either be based on whole structure similarity, or if you've already run one or more of the endpoints for your chemical, it can also perform an analog search based specifically on those alerts that were found by the analog. Next is MetaUltra, which was released in 2016. And MetaUltra is designed to predict what metabolites will form, how likely they are to form. It can also provide physicochemical and toxicological endpoint predictions for those metabolites. And lastly is QSAR Flex, which was released in 2021. As indicated by the name, this platform is designed to be our most flexible software of the 3. Instead of the binary predictions that you will receive from QSAR Ultra, QSAR Flex is actually able to provide continuous quantitative predictions for ecological toxicity and physicochemical properties. And this is also where you'll find most of our nitrosamine prediction tools, including nitrosamine formation prediction, evaluation of nitrosamines for carcinogenicity, and read across for nitrosamines. All of our software packages are available as local installations on your own computer. However, QSAR Flex is now also available as a web app at www.qsarflex.com. So now that I've given you a bit of background on the company itself, I'd now like to go through each one of the software platforms individually and also go through some of the endpoints that are available as part of those software packages. So starting with QSAR Ultra, I'll be going through bacterial mutagenicity, genotoxicity, developmental and reproductive toxicity, which you'll hear me referring to as DART, and skin sensitization. And we're going to start with bacterial mutagenicity. So a little bit of background on this model specifically. So the work on this began in 2014 with the arrival of the ICHM-7. The immediate effects on our research is that we began developing an expert rule-based model, which would be our first, in addition to the statistical models that we had developed previously. We also started working on an expert review tool to go to assist in the review of those QSAR results by our users. We placed an emphasis on regulatory submission readiness in all of these tools that we were building. And then we also worked to build a bigger and more robust model that were built based off of curated data. And all this culminated in the following year in 2015 with the launch of GT Expert, which was our first rule-based model for mutagenicity, as well as Consolidator, which was our expert review tool for ICHM-7. And we have continued to update all of these since then, including in 2017 with the launch of an even bigger mutagenicity model with even larger data sets behind them. So getting into these models themselves. So we have 3 models for mutagenicity. We have two statistical models, which are based on machine learning. And then we have one expert rule-based model, which, again, based on the name, instead of being based on machine learning, this model is based on expert knowledge from literature research. And these models are built and validated as part of an RCA with the US FDA. So the reason that we have two statistical models where we only have the one rule-based model is that FarmB is actually a little more specialized than GT1B. So FarmB focuses more on just pharmaceutical chemicals. And you'll notice that even though it's more focused, it actually has a significantly larger data set that it was trained with than GT1B. And this is because FarmB was built including proprietary chemicals. So these are chemicals where we use them to train the model. We use them to acquire alerts to be used in that model. But then in the actual software itself, those chemicals are no longer present anywhere in the software, again, because they're proprietary. So when you see the report for this model, you'll still be getting those alerts that are derived from those proprietary chemicals, but those chemicals would not be available as analogs in the report. So this next section that I'm going to go through, I'm going to be discussing it specifically in the context of bacterial mutagenicity. However, this also applies to most of the other models within QSAR as well. So QSAR is generally going to give you one of 6 possible results. The most basic and self-explanatory are going to be your positive and negative results. If your compound is already part of the data set for the model, you will get a known positive or negative result. If the model was not able to make a definitive positive or negative call, you'll have an inconclusive result. And if your compound contains some sort of feature that is completely unknown to the training set used for that particular model, that is when you will get an out of domain. So going through the report itself and how it's going to make that final positive, negative, or inconclusive call. So the model starts out by looking for these alerts. And so you have 2 different kinds of alerts. You have these positive alerts, which in this case are structural features that are going to contribute to mutagenicity. And then you have deactivating features, which are marked in blue. These are going to be features that decrease the compound's chances of being mutagenic. And you'll notice for each one of these, they each have an ID number assigned to them. So if we focus in on our one activating feature for this compound, number 7, we move down into where the actual probability is being calculated based on these alerts. So each one of these alerts is assigned a QSAR coefficient, which was determined during the initial, which is determined during the training of the model itself. And so each one of the positive alerts is going to have a positive coefficient associated with it. And each one of these deactivating features is going to have a negative coefficient associated with it. These are all added together in the sum of factors, which is then plugged into the global QSAR equation. And this is where we get our final probability, which in this case is 20.3. Now, how we translate this percentage into this positive or negative call is going to depend on that percentage and how it relates to the gray zone for the model. If the percentage falls below the gray zone, that's where you're going to get this negative call. If the percentage is above the gray zone, that will give you a positive call. And if it falls within this gray zone, that's where you're going to get this inconclusive. And so now you may be wondering, what is this gray zone? So for each model, there is going to be a range of percentages where compounds that fall within this range have kind of a 50-50 chance of being positive or negative. And so because there's not a strong leaning one way or the other within this range of percentages, the model cannot definitively say this is going to be negative, this is going to be positive. Instead, it's going to give you this inconclusive result, which just means that it falls in the middle of the model, and the model is not confident in making that call one way or the other. After the actual calculation of the QSAR probability, you'll also see analogs provided in the report. And so for each one of these analogs, these are going to be compounds from the training set that contain some or all of the alerting features found in your query chemical, and also are overall structurally similar around that alerting feature as well. And so for each one of these, you will see the Ames results from the data for this analog. So all of these have experimental data. And you'll also see a relevance score. So you can see how applicable are these analogs actually to the compound that I'm trying to test. And if your compound is already present in the training set for this particular model, that's where you're going to get your known result. So here we see the first analog is actually labeled as an exact hit with a relevance score of 1.0. And so that means that our query compound is actually in the training set. That's why we're getting a known negative result for this particular compound. And then underneath the analogs, you'll actually see the experimental data for each and every one of those analogs, including your query compound if it's already in the data set. And for each one of these, we'll also give you the literature source for where this data is found if you would like to read further for yourself. If you have a compound that contains a feature that is unknown to the model, all of our models fulfill the OECD requirement to have an applicability domain. And what this means is if you have one of these unknown features, the model is not going to assume that this feature is benign. This could be a mutagenic feature for all we know. And so while the model will still go forward with the calculation of the probability, it's not going to make a final call of positive or negative for this particular compound, because we don't know how the presence of this unknown feature affects or would contribute to this probability. And so instead of making, again, that positive or negative call, that's where we're getting it out of domain in this case instead. So that goes through what you'll generally see in the bacterial mutagenicity model reports. But then I mentioned we also made an expert review tool, and so that's going to be Consolidator. And Consolidator is built specifically for reviewing GT1B Mute and GT Expert. And so what Consolidator is going to do is it's going to look for the alerts that were found by these two models, and then it's going to search its own database for analogs that are not only do they contain those alerts, but they're very structurally similar close to that alert. So it's not looking for whole structure similarity. It's specifically looking for similarity within a certain distance of that alert. And depending on how you set up Consolidator before you run it, you can have it select either 5 analogs, 7, or 9. Here, clearly, I just have 5. And out of those analogs that it found, it is going to have them vote on whether this alert should be confirmed or dismissed. And so if the majority of similar analogs for this alert are negative, then the alert is going to be dismissed based on that majority vote. And so what it's saying here is that while generally this alert might be mutagenic, when it's attached, for example, to a benzene ring, as it is here, it's generally not mutagenic. That benzene ring kind of acts as a deactivating feature for this alert in this particular case. Now, if you're dealing with a case where you have an API and you're checking a bunch of impurities against it, you can use the API as a reference compound. And so what that means is that the API is going to be presumed to be a known negative for mutagenicity, and it's going to see if any of these alerts are shared with that API. In this case, you'll see in this report here, this alert was not shared. However, if it is shared, then that's another way to dismiss this alert as being non-mutagenic. But again, it's not enough for the alert just itself to be shared with the parent compound. It has to be in this same chemical environment in the API as it is in your query compound. So for example, if in the API, if this group was no longer attached to a benzene ring and it was instead attached to an alkyl chain instead, then it can no longer be dismissed because it's in a completely different chemical environment. Now, the role that Consolidator serves, it enables interactive and transparent evaluation of those alerts and analog provided in the report here. And it allows you to review and challenge those alerts, which is going to be essential for justifying these QSAR results to regulators. And then lastly, it improves the defensibility of these calls by letting you trace the alerts back to this source data. So again, for each and every one of these analogs that we provide, we'll also provide the experimental details behind each one of these results, as well as the literature sources for those. So that covers bacterial mutagenicity. So now we're going to move on to genotoxicity. So we have several different models for gene tox. However, the purpose for all of these is to assess chemicals for their potential to cause genetic damage. The first set of models we have are for the Chinese hamster cell lines. We have both the CHO and CHL lines. In addition to those, we also have the combined model, which combines the data for both of these cell lines. And the benefit of having this combined model is going to be that because you have this bigger data set, you have a lower chance of having out-of-domain calls. And then it can also help to clarify if you have conflicting results between the two single cell lines. We also have a mouse micronucleus test, which is our single in vivo endpoint for gene tox. And then we also have the mouse lymphoma assays, both activated and unactivated, for detecting indirect and direct acting toxins, respectively. We'll go through a brief case study for this endpoint with rocuronium bromide. So looking at these using the CHL and CHO endpoints. So for CHL, we had an out-of-domain, and for CHO, we're getting an inconclusive. So clearly we're not getting a whole lot of helpful data from these two individual cell lines for this compound. If we look into those analogs that are included in the report for the CHO result, we see that first of all of these compounds seem to be relatively similar to our query compound, and also out of these first four, which are all pretty similar in terms of their relevance scores, out of these first four, 3 of them are positive. So maybe instead of inconclusive, we can say that this call should be positive instead. And then we can confirm this by running the combined cell line, which also gives us a positive result, confirming that that should be the final call for this compound. Next, we'll move on to DART. So for DART, we have a number of models. However, they can be divided into generally into 2 groups. So the first being developmental, where we have the fetal dysmorphogenesis model and newborn behavioral toxicity, as well as the reproductive models focusing more on the parents, both for female and male fertility, and then also focusing in on sperm toxicity specifically. For each of these models, we have both the mouse and rat models. And then for fetal dysmorphogenesis, We also have a rabbit model. Across all of these, the rat models are going to have the larger data sets of the two, meaning that the rat model is more likely to give you a known positive or known negative, and it's less likely to give you an out-of-domain result. So again, going through a case study for this endpoint with alprazolam. This is for all of those models I showed on the previous page. You see for most of them, we're getting negatives or even known negatives. But for a few, particularly the mouse models, we're either getting inconclusives or a positive. But then you'll notice on this expert conclusion column, so this is the conclusion based on human review, we're saying that all of these should actually be negative. So why is that? Well, if we start with the sperm toxicity model where we got a positive out of the mouse model, if we look at those analogs again, so initially we might say that, okay, out of the first four, 3 of the 4 are positive, so maybe we should have that positive call. But if you look into those relevance scores and even just look at the structures themselves, you'll see that this first chemical, midazolam, is actually much more relevant than the other 3, meaning that we should give this call as a read across evaluation. We should give this more weight than these other 3. And so based on the results for this very, very similar analog, the call for the sperm toxicity in mouse should maybe be negative instead because we have such a strong analog. And that's how we're coming to the negative conclusion for sperm toxicity. And I don't have time to go through it, but this is the exact same reason why we're making the negative call for the male fertility as well. Looking into newborn behavior. So for this one, we don't have that super similar analog in the mouse model, unfortunately. However, if we look between the alerts found by the rat model versus the mouse model, both of them are identifying this benzene ring as an alert. However, the rat model is giving us that known negative, and that known negative is going to be very strong, especially because both of these models, despite being in animals, these are both looking at that same endpoint. So because we have both a known negative and both models are finding that same, they're agreeing on what the alerting feature is, we're going to rule that as a negative call for newborn behavior overall. And then lastly, we'll move on to skin sensitization. So skin sensitization is trying to identify chemicals that will elicit either an allergic response, will cause irritation, or will be destructive to the skin or eyes. And so we have several models for these. We have the LLNA model for the local lymph node assay. We have non-LLNA. We have our second expert rule-based model. We have models for skin irritation and corrosion. And then we also have a couple models for eye irritation and corrosion as well. And so particularly for chemicals that can elicit an allergic response, it can be critical to not only give, a positive or a negative result, which is typically what you're going to get from Case Ultra, but actually getting an estimation of what the specific potency of that chemical is going to be. So how we do this in Case Ultra is, in addition to the base LLNA model, we also have these categorical models. And so how these categories are determined is each of these is associated with a certain concentration of the chemical that is required to elicit a 3-fold increase in that lymph node cell proliferation, with category 2 being the strongest. And for our models, that also we also include category 1 as part of category 2, just for the sake of the model building. And it goes all the way down to category 4, which would be those weakest sensitizers. And so clearly the arrangement of these models is maybe a little bit more complex than what I've shown before. So what is the workflow for using especially these sensitization models in order to make a prediction? So our recommendation is to start with the expert rule-based model, which again is going to give you those mechanistic explanations for why certain functional groups are going to be sensitizers, as well as using the non-LLNA model. If both of these are negative, then we rule the compound as being not a sensitizer. However, if either or both of them are positive, or you get an out of domain for either of the models, then you move on to the LLNA model. If this is negative, then we accept that as the final conclusion. However, if it is positive, then we move on to those categorical models. And so how we use the results from those categorical models to determine the final what category of sensitizer our query chemical is we take the positive result from the most conservative prediction. And so that means if for a certain chemical, all three of these are giving us positive results, then it's automatically a category 2. If we are getting negative results for these latter two and a positive for 2, it's still going to be considered that strong sensitizer. So looking at a case study in the form of ethanol. So starting with, again, that expert rule-based model and the non-LLNA. So the expert rule-based model is giving us a positive result based on these alerting features. And then for non-LLNA, we're getting an inconclusive. So if, again, we go into those analogs and look at what the results for those most similar chemicals are, first of all, we can see that we've got pretty good similarity across these different compounds, both in terms of like how similar they are to our query compound and how similar they are to each other. And if we were to take a majority vote from these, it would come out as negative. However, if because we got that positive result from the expert model, we are now going to run the LLNA model. And so this comes out as a known positive, meaning that this chemical is part of the data set for the LLNA model. And because we got a positive there, we move on to determining what category of sensitizer it is. And here we got a positive for 4, a negative for category 3, and then a known positive for category 2. And so this is the result that we're going to go with, making it a category 2, a strong sensitizer. So that was a quick run through of 4 of our endpoints. However, there are many more available through Case Ultra, including acute toxicity, adverse effects, carcinogenicity, cardiotoxicity. And each one of these models, or each one of these endpoints, will have several models associated with it as well. If you would like to try your hand at building your own model or modifying one of our models, we do give you the opportunity to do so. If you have a sufficient number of chemicals, you do have the option to build a completely new model simply by clicking the New Model button. It will allow you to input your experimental data, including units, the units that your results are in. And this model will be built in anywhere from minutes to a few hours, depending on how big of a data set you gave Case Ultra to work with. If you just want to modify one of the models that we provide, so for example, maybe you have a lot of chemicals that have a particular feature that's coming up as an unknown fragment in one of the models, you can add your own chemicals with experimental data to our training chemicals and use that to rebuild the model and hopefully decrease those out of domains in your chemicals that might have that novel feature that wouldn't otherwise be part of the data set. And it's important to note that for these new models that you're building, whether you're adding chemicals or building a completely new model, these will all exist exclusively on your own machine. These are not going to be sent to us. They're not going to be uploaded to the cloud somewhere. So that if you're using proprietary chemicals and building these models stay entirely with you, entirely on your computer. However, we do also offer the service where if you would like us to, we can also build customized models for you, assuming that you have a sufficiently large data set as well. And as part of that, we can also use data from our data sharing consortium, which includes other proprietary chemicals as well, where again, while those chemicals themselves are not going to be shared, the alerts used or the alerts derived from those chemicals will appear in the models. So that covers Case Ultra. I'm going to now quickly go through Meta Ultra, which is our metabolite prediction tool. So Meta Ultra uses QSAR predictions as well as a database search to generate both potential metabolites as well as known metabolites that are going to be part of that database. And it can also evaluate those metabolites and predict their physicochemical properties as well as toxicological endpoints using models from Case Ultra. The report for Meta Ultra is going to be rather simplistic, where you're always going to see the parent compound in the top right. This first column are going to be your phase 1 metabolites, and then you'll see the phase 2 metabolites that are derived from those phase 1. If we zoom in on this metabolite and look at what exact information you're getting underneath that structure there, the red half circles you see beneath each one of the structures indicate or are correlated with the QSAR calculated probability of that metabolite forming. So you'll notice that for each of these, they kind of begin to fade out, typically as you get further down into these phase 2 metabolites. Underneath that is the metabolic mechanism that caused this particular compound to form from that parent compound. If your compound is an experimentally known metabolite, we will tell you. And if it's a major product, or if it is a major metabolite, we will let you know that as well. And then the last piece of information that we're showing is the QSAR calculated probability of this metabolite forming, which again correlates with the red half circle shown at the top there. If you want to look deeper into this metabolite, you can either right or left click on it. Left clicking will give you the sites of metabolism on the parent compound, as well as what structural features around the site of metabolism affected that final QSAR probability score. And then if you right click on the compound, this is where you'll find those physicochemical properties, as well as the endpoints for any Case Ultra models that you have available. So that was a brief overview of Meta Ultra. Now we're going to move on to QSAR Flex. So QSAR Flex is designed to be, again, much more flexible than our other software. And namely, this comes in the form of being able to make continuous predictions for certain properties and certain endpoints. So if you're looking for something like an LC50, a boiling point, water solubility, positive or negative really is not going to cut it in terms of an output. You need an actual number, and QSAR Flex is going to be able to actually give you that quantitative prediction. In addition to these continuous ecotox and physicochemical property models, this is also where our nitrosamine suite is, and this includes 4 separate tools. So the first of those is a cross-similarity tool, which looks for pairwise similarity between a list of nitrosamines that you provide. We have our older surrogate search tool, which uses read across to find relevant analogs for your query nitrosamines that will then have experimental data that you can use. We have our CPCA prediction tool, which calculates carcinogenic potency as well as the acceptable intake for those nitrosamines. And then we have our N-nitrization tool, which predicts the likelihood of nitrization of an amine-containing compound. So of these, the surrogate search is going to be our oldest tool, and so this is purely a read-across tool. It was developed as part of an RCA with CEDAR back in 2021. And so this is going to find compounds that have Ames data that are structurally similar to the compound that you're trying to test. And so it's going to make that similar, it's going to search for similarity based both on the whole structure as well as the alert environment itself, specifically surrounding that nitrosamine group, because that's going to be what's most critical for determining how carcinogenic this nitrosamine could possibly be. We followed this up a couple years later with the CPCA prediction tool. And so this takes you through the CPCA workflow to determine what potency category a nitrosamine should be categorized into, with the categories being 1 through 5. And each one of those is also going to have an acceptable daily intake associated with it. This tool also includes a surrogate search tool. However, instead of searching based on whole structure similarity or just alert environment similarity, this tool is going to search specifically based on based on similar CPCA scores to your parent compound. And if we have the regulatory AIs for your query compound, or that we'll have for those AIs, we'll give you those so that you can maybe make you can argue for or maybe even against the AI and the category that was produced by the CPCA. And then lastly, we have our nitrization tool. So instead of evaluating nitrosamines, the goal of this tool is to determine how likely that nitrosamine is to form in the first place. And so this tool is based on a series of expert rules. And so we'll show you in the report exactly how we calculate the formation score for that nitrosamine. So here for tertiary amines, we assign tertiary amines a score of -4, meaning that they're going to be less likely to form. If your nitrosamine is going to require a leaving group that's larger than ethyl, which in this case to form NDMA, we have to lose this whole big group. That's an additional score of -2. And so that's how you respectively get these scores of -4 and -6, with the lower the number corresponding with a lower potential of formation. For each of the predicted nitrosamines, we'll give you the CPCA prediction coming straight from that tool that I showed on the previous slide. If we have regulatory AIs for a compound or carcinogenicity data, that will also be given to you, as well as computing the physicochemical properties or providing the experimental values if we have those available, so you can compare them to the parent compound and determine how feasible it's going to be to separate the nitrosamine from the parent compound, if that's going to be something that you need to do. And then, as always, we provide analogs for all of our data as well. So if your compound is already part of our nitrization database, we will give you the percent yield from that experiment, as well as those experimental conditions. And if your compound is not part of that database yet, then we will give you, again, those closest analogs and the experimental data and yields for those instead. And if you'd like more information on this tool, we just put out a paper on this earlier this year, and it is open access, so it's going to be free to read. So that was kind of a whirlwind tour of pretty much all of the tools that we have available, as well as a number of the endpoints within those tools. So what do we have coming up next? Well, that's going to be oral bioavailability. And so with this, we're looking at what percentage of a substance is actually going to enter the body circulation in order to have some sort of an effect on the body. And so as part of this new set of tools, we've developed a manually curated database of about 1,600 compounds, which is then used to build a statistical QSAR model based on physicochemical properties of the query compound, as well as fragments that were pulled from this curated database that it was trained on. We also offer a read-across tool that allows you to compare properties from compounds that have known data to your query compound. And then we also have a simple expert rule-based model that's based on molecular weight and log P. So quickly going through each one of these tools individually. So the statistical model is going to tell you which bioavailability class your compound is likely to fall into, ranging from very low to high. And again, this prediction is going to be based on the physical-chemical properties, so log P, water solubility, vapor pressure, as well as fragment descriptors, similar to what I was showing in those case ultra reports. And so this is a sample prediction that you would get out of this tool. So not only is it giving you what class it thinks this compound will most likely fall into, it's giving you how confident it is that this is the correct class for this compound. So in this case, we have almost a 70% chance of this compound being falling into medium, meaning that it has a bioavailability between 50 and 90%. But we still have double-digit percentages for the low and high classes. And so you might have a case where maybe it's 50-50 between low and medium. So in that case, the model isn't really giving you a very definitive answer. And there, you might want to look into some analogs, the same as what we were doing when we got those inconclusive results in Case Ultra. And so the first of these tools is going to compare the physical-chemical properties for your target compound, for which you do not know the bioavailability, with compounds for which that data is known. And so here you see our target compound in gray, overlaid with the properties for the analog in blue. And this is the same information, just displayed a different way in this star plot on the right here, where the features that have a longer leg, these are going to be features that have a stronger contribution to that exposure value. And so for this one, we have pretty good similarity, although we do have one feature that is significantly higher, that being the total polar surface area. So where in the previous, for the QSAR model, we were saying it was medium. This, our closest analog says it might have a high, should be rated in the high class instead, but maybe not, considering that we one of the features is much, much greater in the analog than it is in our target compound. The second read across feature we show, and this is just also in support of how well is your compound covered by the data set for this model. You can look at both how similar are these analogs structurally to your compound. So here for this first one, we have similarities ranging from 0.92 down to 0.84. And how does the bioavailability for those compounds vary between them as compared to your structure? And so in this case, these are all pretty tightly grouped in addition to being pretty structurally similar. So that gives us a pretty low uncertainty as to what class we should rule this compound in. Now, if you have the case where your similarity scores are a bit lower and then the bioavailability is kind of all across the board, that's going to increase your uncertainty for the predicted bioavailability for that particular compound. And so this is just giving transparency for how confident can you be in the prediction made by these models. And then finally, if all else fails, we also do have a very simplistic model based on published literature that simply takes into account molecular weight and log P and makes a prediction of bioavailability based just on those two features. And so that's kind of what's next for us. Now we can discuss what's next for you. If you have any questions or you'd like to give the software a try for yourself, you can get into contact with us at multicase.com/contact. Or if you're in one of the countries or regions covered by one of our local distributors, we would also encourage you to get into contact with them, and they would also be happy to set you up with a trial or demo or to answer any questions you may have. And with that, That brings me to the end of my presentation, and I'd love to open up the floor for questions. Thank you so much for your attention.
Hi, everyone. This is Gianna from MultiCASE. I'm here with Crystal and Rustam, along with others from our MultiCASE team. And we'd like to open it up for questions. As Crystal invited you earlier, if you do have any questions, you can always type them into the questions panel. Or if you prefer to verbalize your question, there is an option to raise your hand and we can bring you off mute. There were a few questions that came in during the presentation, so I'll address those first. And if there's any additional questions, we're happy to answer them for you tonight. So one of the questions that came in during the presentation is with regard to the mutagenicity model suite, in particular, the second statistical model containing proprietary information known as farm-bmut. An attendee was asking whether or not it's possible to highlight analogs that are produced by the farm-bmut model. Since the structure is hidden, how accepted is that by a regulatory agency when you're trying to highlight different structures, whether that be analog or alerts or deactivating features that are identified by that model?
I can answer that. It is acceptable as a weight of evidence additional piece. You cannot rely your response to regulatory agency based entirely on farm units, but you can contribute the results from farm unit in addition to your classical GT1 unit and GT expert answers. The classical example would be if these two models do not give you any real answer and consolidator doesn't help. No call. If you test it with farm-bmut and it comes as negative, or if the reason of no call was out of domain and it's not out of domain inside farm-bmut due to the better structural coverage, that would be a very convincing piece of evidence. And yes, since at least half of the models of the chemicals within farm-bmut are not just hidden, they are removed from the model. They're not accessible in any way. Not all of the analogs will be available. That's inevitable. We want to have a high quality model. We cannot disclose proprietary chemicals. That's the price to pay.
Okay. Hopefully that answers your question. If you have any follow-up questions, let us know. Another question that came in was with regard to the reliability of the tools and how to determine their prediction accuracy.
Unless Crystal wants to answer it, I can also chime in. We
Yeah,
perform
go
all
ahead.
validation by the rules. Whatever is recommended by OCD, cross validations, bootstrap, while sampling, etc. And we also, at least for most of our models, when we have extra data, we do provide external validation results. It's all reflected in QMRF documents, which are available for every model. That's how you can judge the reliability of those predictions.
Okay, thank you, Rustam. Someone is asking, is this software the same as Nexus? Do you use Derek and Sarah database or any other? Is the software and data based and approved by the US FDA? So I guess two questions.
The only thing which is the same between different QSAR models is the technology behind it in general, QSAR. Each of the vendors using their own algorithms, each of the vendors using their own data to which they have access. Part of this data might be provided by US FDA within not data sharing, this agreement within research cooperation agreements. Part of it, not all. So fraction of the data would be shared between different vendors, the another fraction would not. That will result in physically different models and in not absolutely, but quite frequently different predictions. Our competitors might capture something which we don't have, or we can capture which they don't have. So if you expect to have identical predictions between, let's say, their products and our products, you would be very mistaken. But that's why US FDA, for example, using several vendors as complementary tools. Whatever is not captured by one vendor will be captured by another. That's the answer.
Thanks, Rustam. Another question coming in is with regard to call confidence. And I know Crystal supplied a few case studies. I guess it's at a high level, right?
Gianna, your mic is going out.
Is this okay?
Yep, that sounds better.
I talk too
Okay.
much. Let Crystal answer.
Someone is asking, how can we determine confidence of QSAR output?
So is that the whole question, determining the confidence?
Yes.
Okay, so I think that was already answered a little bit by Rustam, where we do provide some of that information as far as like the overall confidence you can have in the model as part of the QMRF that's available for each one of those models. And then the other part of the confidence, I think, is going to come from when you are looking into that report itself. So the percentage that you're going to be given as that QSAR prediction, the closer you are to either 0 or 100%. So that's going to give you some more confidence in that call, the farther you are away from that gray zone. And then also looking into the analogs that are provided as part of that report. If when you're looking into those analogs and you're looking into the relevance scores that are provided for those analogs, if all of those relevance scores are relatively high, then you have a good idea that your compound has quite a bit of experimental data from similar compounds that were used to train this model. However, if those analogs have very low relevance, maybe you're looking at scores of 0.2 or maybe even 0.1, then maybe even if you have a very either high or low percentage for that QSAR calculated probability, because those analogs are so dissimilar from your compound, then that might mean that you should have less confidence in the call that's being made based on those model. The call that's being made by that model. So kind of a short answer, it's a combination. It's going to be a combination of the QMRF, exactly what percentage you're getting from that QSAR model, and then how similar are those analogs coming from the data set? How similar are those to your query compound?
In addition to that, in some cases, we actually able to provide the confidence as a single-digit number, just a number for some of the most recent advanced QSAR Flex models. This technology is fairly new, so it will be expanded to some other cases, and hopefully, rather sooner than later, we'll have the same technique available for all other models. And then, in addition to whatever Crystal mentioned, you will simply have a confidence number. This call, whatever it is, negative or positive, with all these analogs, alerts, etc., has this much of confidence. That is already possible for some of the models, not for all of them, but soon enough.
Okay, great. So I think we came to the end of our questions for tonight, but if anyone has any lingering questions later on, there's always an opportunity to reach out to us either through email or through our website. As Crystal mentioned, we have several distributors crossing multiple regions globally, so feel free to connect with any of them, or if you don't know how, we can always get in touch with us and we'll help you connect with them. And as Rustam mentioned, we'll be at a few conferences in the next few months. So if you're planning to attend, you can always connect with us in person. So more opportunities to stay in touch with us and find out what we're up to. In the meanwhile, stay tuned because we will be hosting more webinars on case studies specific to not only mutagenicity, but endpoints outside of mutagenicity, similar to what Crystal has presented tonight. So I guess I'll leave it at that. And thank you all for taking the time to attend tonight's presentation. Hopefully we'll be in touch soon.
Thank you all.
Thanks, everyone.
Thank you.